In this video, we're going to continue our discussion of microbial diseases, continuing with those bacterial diseases. And we're going to look at the next four on our list with diphtheria, tuberculosis, botulism, and peptic ulcers. So let's begin with diphtheria. Diphtheria was one of those diseases that was highly dreaded. However, through the years and the modern medicine and vaccination processes, we've been able to bring the cases of diphtheria down greatly and get it kind of under control. But that's because of that, again, the vaccination process and realizing that diphtheria is part of that DTaP vaccination. So let's talk a little bit about what diphtheria is. Diphtheria is going to be caused by a bacterium known as the Corynebacterium diphtheria. It's a gram-positive bacterium. As you can kind of see over here in this picture, it's going to be a club-shaped bacterium. It is an obligate aerobic, so it's going to have to be in areas where we're going to have some oxygen. And it does, sometimes you'll hear it say that it's a palisade, and that's because of the fact of how this bacterium tends to clump together. Now, when we talk about diphtheria and bacteria, it produces what's known as a cytotoxin. Okay, and again, cytotoxin means that it's going to be a toxin that's specific for cellular processes, and in particular here it's going to inhibit translation in cells of the host. Now remember translation, so you go transcription and translation, so that's going to have an impact on protein synthesis. And in particular, the cytotoxins is that classic AB toxin that we discussed in a previous unit, of which it's going to have the binding, which makes it specific for particular tissues, and then the A part of the toxin is the active substance that is actually going to, in this case, it's going to help shut down protein synthesis by inhibiting translation. So how does diphtheria work? Diphtheria is going to come into the body through respiratory droplets. So transmission is through respiratory droplets, even though it can be through some fomites. Now remember the term fomite from previous units. That just means that's an inanimate object that's used to help transmit or transport a disease. So because it's coming in through respiratory, it's generally going to start out with a local infection in the pharynx or the throat region. And then once it's in there, so it becomes established, starts that multiplication, then it's going to start to release its toxin as part of those steps in pathogenicity. As it releases that toxin, because it's a cytotoxin, it's going to start to cause those cells to die and become necrotic. Now that's part of what makes it a tall tale symptom, is the fact that you know it's diphtheria, because what forms is this very thick hard tissue or membrane that's actually kind of a greenish gray in color, and it's referred to as a pseudo membrane. And so you'll see that on the tonsils or the throat, and we're going to get to that, and you'll see a picture of it here in just a minute. So we get the pseudomembrane, but remember, because it's producing the toxins, that as it continues to produce the toxins and as this necrotic tissue is forming, then those toxins can get now into the bloodstream and the fluids. And so then it has the potential to get even into more tissues and into deeper tissues, spreading amongst the body. So it goes from being a local infection either to a secondary or even potentially systemic, and leading to more serious illnesses, and if not treated, to death. So again, see here in this picture, you can see this very kind of thick membrane tissue that's kind of got a greenish gray. That is that pseudomembrane. So you'll notice that here too, that you see all that redness that's occurring, because again of that cytotoxin that's being released, and it's going to then cause somebody, and it's attacking the throat. So just think, it's going to be the same kind of symptoms that you would expect in any sore throat. So you're going to have hoarseness, and then you're going to have fevers, and you're going to have swollen glands, all because of this. So How are we going to treat diphtheria? We're going to treat it first because it's a bacterial infection. We're going to want to get rid of the bacteria, so we're going to have to use antibiotics such as penicillin or erythromycin. However, because it's also using a toxin to do damage, don't forget, and especially because this is that AB toxin, we're going to have to neutralize out that toxin, because if you just clear the bacteria, it's the toxin that's doing the damage. So by the time you see this, that means there's plenty of toxins in the body. So we're going to have to get rid of it. So you're also going to have to administer an antitoxin. And the sooner you can get that antitoxin administered, the better chance that patient has of a full recovery. So again, this used to be considered to be an extremely important disease. It was caused a lot of concern, but now because of the vaccination process, and again with it's part of that DTaP, then we're able to keep this one under control. So now let's look at another disease that's specific to bacteria, and that is tuberculosis. Again, this is one that you're all very familiar with, but we're going to get into some of the more details in this case. Now, when we talk about tuberculosis, tuberculosis is caused by the bacteria Mycobacterium tuberculosis. Now, it is a mycobacterium. Now, remember from previous chapters when we were talking about bacteria, Mycobacterium is an acid-fast bacterium. Now, what does it mean to be acid-fast? That means its cell wall structure is going to be different than the gram-positive and the gram-negative with all those peptidoglycans. In the case of Mycobacterium, they're going to have that mycolic acid in that cell wall. And the mycolic acid in the cell wall is part of what gives this bacterium its virulence factor. Because remember, it's that waxy substance. So if you're taking a cell and you're kind of wrapping it in wax, it's going to do two things. One, it's going to help it be antimicrobial effects of the host. It's going to protect that cell from that. And the second thing is it's going to make it be a slow grower. And so that means that we're going to have to think about that when treating these types of bacterial infections. So when you talk about Mycobacterium tuberculosis, again, that mycolic acid is one of those main virulence factors. Because of that waxy layer in its cell wall, it's going to make it impermeable again to those microbial agents that the host is producing, but it's also going to make it be a slow grower. Now, another piece of the puzzle for the Mycobacterium tuberculosis is that it's going to interfere with normal macrophage abilities. It's going to prevent a macrophage from actually, when it engulfs this microbe, instead of destroying it, the microbe is going to be able to replicate and divide inside that macrophage. So it's going to help it to basically hide from other immune responses that the host is mounting and trying to get rid of it. Now, through part of its pathogenicity, what you're going to find too is that in a Mycobacterium tuberculosis, that it gets into the lungs and it's going to form what we call a tubercle, which is really a granuloma, but it's because it's in the lung, we refer to it as a tubercle. Okay, and so this is what's going to harbor that tuberculosis, and then it's going to actually hide it for a while. So let's look a little bit at that pathogenicity. Now, recognize one of the things in tuberculosis is that there's basically three types. You have a primary, secondary, and you have a disseminated tuberculosis. But when somebody gets TB, most of them are going to be in a latent or inactive form. Only about 10% of those develop into the active form of the disease. So let's talk a little bit about the how this tuberculosis works. What is its pathogenicity? So first, when the tuberculosis bacteria comes in, it's going to enter in through the respiratory tract, through aerosols, so the droplets. It's going to get in, it's going to get into the alveoli, and it's going to begin to multiply. But as soon as it gets into the body, it has breached those host defense mechanisms. So the host defense is going to be like, wait a minute, we got to get our cellular responses in there. And think about it. What are the first cells that come in? Those phagocytic cells, and in particular, the alveolar macrophages. So the macrophages in the alveoli are going to come in and they're going to phagocytize the bacteria. But like we've already mentioned, the bacteria are going to inhibit those macrophages from being able to actually destroy it. Okay, so because of that, they're going to be able to get into that macrophage and they're going to be able to replicate. But because the macrophage has already been infected, then it's going to start acting like an APC and it's going to start calling helper T cells in there. And then that's going to help to start that inflammatory response and call other macrophages and other immune cells into the area. Now, why is this all important? Because as you're calling in the macrophages and the other immune cells, these are what are going to start surrounding that bacteria as it's replicating and dividing. And this is what's going to end up forming what we call that granuloma, or in the case, because of the case it's in the lungs and it's being formed because of tuberculosis, we call it a tubercle. So you might see, depending on where you look, what term is used there. But then that forms within our lungs. And once you form that granuloma and it becomes this very hard substance, and you can see that on x-rays and stuff, but at this point it protects the body because it has encapsulated everything, for lack of a better term there. And so when you have the granuloma, the person's kind of in a latent state of that infection and they'll stay there. But ultimately that granuloma can burst open and then the bacteria is now released and it leads to that inflammatory response again, and so it just keeps building up. So again, one thing to recognize about tuberculosis is that because of the formation of the granuloma and because of bringing all those cells in, the body's host defense mechanism is able to keep this disease under control for a long period of time. So somebody they may test positive for TB, but they may be in a dormant state of it, as I said, latent state of it. And only about 10 or 15% of that are going to actually turn into an active infection. So just kind of a quick drawing. So transmission, of course, is from that respiratory droplets. And so once you get that bacterium into the alveoli of the lungs, then it gets into the macrophages. Then even though it's in the macrophages, remember the bacterium inhibits that macrophage from carrying out its normal process of ingesting the bacteria, fusing that phagolysosome with the lysosome, and then having those enzymes actually kill the bacteria. It prevents that normal process, but instead you can see here that the bacterium is staying inside those macrophages and we're calling all the other immune cells in there and keep surrounding it with various immune cells, and then we form this very large granuloma, which is a hard complex, and then it kind of keeps everything dormant for a while and it stays there until ultimately that granuloma will burst open. It becomes necrotic and burst open, and then we're able to release lots of different bacteria in, and then we repeat the process. So how do we treat tuberculosis? Okay, again, a lot of times it's going to be a latent tuberculosis and people aren't actually exhibiting symptoms. Only those with the active TB are going to be, you're going to start seeing them cough and you're going to have fatigue and sweat and things like that. So if you'll recall, we talked about the fact that the mycolic acid provided those cells a protective mechanisms and it helped defend them from a lot of the antimicrobial responses that the host defense mechanisms will mount. But the other thing about it is because it's got that waxy coat going around that microbe, it means that microbe is going to be growing at a very slow pace. So it has a slow generation time. So if it's got a slow generation time, that means it's going to take a long time for us to combat it. And so it's going to usually take a long period of being on antibiotics for a patient. And you can't just have one antibiotic over that length of period of time, because then we'll allow that bacteria to possibly build up a resistance. So as you'll see here, it's going to be a combination of various drugs and antibiotics to be able to help a patient who has tuberculosis. Now, how do we determine if somebody has tuberculosis? We obviously do that Mantox skin test, and we're looking for that. You can also look for chest x-rays. They can take blood samples or sputum samples and start looking for the presence of acid-fast bacterium. Okay, so let's switch gears from tuberculosis and talk a little bit about botulism. Now, botulism is going to be a fairly serious disease because of the toxin that is being secreted. And in this case, it's a botulinum toxin. So the toxin is going to be the virulence factor for this disease. And it's one of the most potent toxins known out there. And unlike the other diseases that we've discussed, which are respiratory, skin, etc., this one is going to attack the nervous system. Now, botulism is a little bit different because it's got different ways for which a person can come in contact or get that botulinum toxin into the body. One is from foodborne. Now, foodborne means that the food itself, when somebody ingests it, has been contaminated with that toxin because the microbe has been in the food, it has multiplied, divided, and released the toxin. So people can actually just ingest the toxin through the food. It can be from a wound infection, of which the bacteria then enters into the body, establishes itself, and releases the toxin. In the case of infants, we give it its own name, and we call it infant botulism because it is generally going to be due to the consumption of honey, because the honey will have the botulinum toxin in it. And even though adults may have the ability to fight against small amounts that may be found in honey, infants haven't had a chance to build their immunity to it, to build that memory, and so therefore they are often going to, if consumption of honey, they can have botulism side effects. So that's why a lot of pediatricians and stuff will say in documentation will say not to give infants honey. And then with the rapid rise of Botox treatment, we're beginning to see some botulism toxicity can occur to that as well. So when we talk about botulism, what's the causative agent? Well, it's due to a Clostridium botulinum bacteria. And again, it's a gram-positive endospore bacterium. So an endospore making it unique as well. So in this picture over here, you can see the endospore either within the bacterium, and then of course there are some that are free-floating as well. So it's oftentimes going to be found in soils and the digestive tract of animals and things like that. So again, there is a bacterium, Clostridium botulinum, which is going to be responsible for botulism. Now, again, what is the virulence factor? Botulism is a toxicosis, meaning it is directly related to a toxin infestation or ingestion, okay? And it is particularly due to the botulinum toxin or exotoxin. Now, the toxin itself is classified as a neurotoxin. Why? Because it attacks the nervous system. In particular, it's going to prevent the nervous cells from releasing the neurotransmitter acetylcholine. And you probably remember that acetylcholine is what then leads the muscles to contract. So it gets released from a neuron, and then acetylcholine will signal for muscles to contract. So if a muscle can't contract, then that means it's going to be in a flaccid state. So what happens? How does this occur? So there's different ways. So let's look first at what the foodborne pathogen is for botulism. So in this case, contaminated food is digested. So that means the bacteria is in the food, the bacterium has released the toxin, and you have ingested that toxin. So through the consumption of food with toxins, the toxin gets into the GI tract and then it gets absorbed. And so then that neurotoxin gets absorbed into the bloodstream and gets into the central nervous system. So once it gets into the central nervous system, as already mentioned, it's going to interfere with the release of acetylcholine. And so therefore, if it's interfering with acetylcholine, then the neuron can't release that neurotransmitter, and then muscles are not going to be able to contract. So if they're not able to contract, they remain floppy or flaccid, but it's still a form of paralysis. If you're not able to move the muscle, you are going to be paralyzed, and in this case, it's a flaccid paralysis. And in particular, where this is going to become important is in the respiratory muscles, and in particular that diaphragm. Now, again, we talked about botulism has more than one entry point. So in some cases, we can get it through a wound because remember, the Clostridium bacteria is often found in soil. So if you're outside working in the garden or you're outside walking around barefooted and you cut yourself and you get the dirt in it, you might have a chance of getting Clostridium. So in that case, what happens is the toxicosis occurs because you've gotten the bacteria into the host. The bacteria then replicates, divides, and then it starts to secrete the toxin. And so once that toxin gets secreted into the blood, it's the same pathway that occurred with the foodborne toxin. So in the infant botulism, again, it's going to be an ingestion of the honey, and so you're going to get the toxins that way. And because infants just haven't had time to build up that memory, it's going to have a greater impact on them. So it'll be, sometimes you'll hear it called floppy baby syndrome because of that flaccid paralysis that occurs. So because of the fact that it's a neurotoxin, then one's going to expect people to have headaches and blurred vision, difficulty speaking and swallowing because we're shutting down muscle contraction abilities. And in particular, as I said, one of the biggest ones to be worried about is that diaphragm, and it's going to lead to respiratory failure if you don't treat this rapidly, because if you can't breathe, it's going to lead to death. So how do we treat it? The biggest thing that we can do is you got to get rid of the toxins, because the bacterium itself is doing no harm. It is the toxin that it releases that is causing the major damage. So the first thing it has to be is large doses of antitoxins immediately to help neutralize out that toxin and prevent further damage or even death. Okay, so the last one for this video we're going to look at is the peptic ulcers. Now, ulcers just mean sores or open sores, and in this case peptic is referring to the fact that we're looking at that stomach or gastric region. So in particular, the peptic ulcers are going to be sores that are in that gastric region, and it can even go down into the duodenum of the small intestines. Now, for a long time back in my generation and which is probably your parents' generation, from some of you, grandparents' generation, that ulcers were considered to be, oh, you're producing too much stomach acid because of the stress of your lifestyle or the alcohol consumptions or particular foods or smoking. And it has since been learned that that is not the case, that peptic ulcers are caused by bacterium. So the real causative agent for peptic ulcers is something known as Helicobacter pylori. So H. pylori is the causative agent for peptic ulcers. So we do know that, but there's still some pieces of ulcers that aren't all known. But do recognize here is a picture of the pylori bacterium, and as you can see, it's flagellated, so that gives it motility, which is part of its virulence factor because it can move around. So it's a flagellated gram-negative, and see how it's kind of got a curve to it, so we say it's Vibrio-shaped bacterium. Now you may recognize this picture because I used it in a previous unit when we were looking at, hey, the general discussion of how bacteria can release various toxins and enzymes to protect themselves against host defense mechanisms. And the Helicobacter pylori is one that has all kinds of defense mechanisms and ways to induce damage or harm to its host. Now, we've already mentioned the fact that it's flagellated, so the fact that those flagella can help it move is a virulence factor. It does also release several different enzymes and toxins. One of the biggest enzymes to recognize is urease, because urease is going to be able to go in and break down uric acids and stuff into ammonia, so it's going to help to neutralize out those stomach acids. It's also, because it's gram negative, it's got LPS on its surface over here, and then it's going to have various exotoxins and then other enzymes such as mucinase, protease, and lipases, which are going to actually go in and do damage to that mucosal membrane. So let's look a little bit here at the pathogenicity. So you get the bacterium in, and this is still kind of unknown a little bit, if it's always food or how people get in. Is it oral fecal, and how do we get that bacteria in? But the bacteria is going to get into the gastric system. It's going to get in and it's going to go straight to that mucous membrane. Now, when it gets there, it's going to start to release that urease enzyme, and that's going to neutralize by producing ammonia. Now remember, if the acid in the stomach is acidic, ammonia is basic, so we start to neutralize out the gastric juices. And when you neutralize out the gastric juices, you are then going to give that bacterium a better chance to replicate and divide. So then it comes in, it escapes that first defense there, it's able to replicate and divide, and it's doing so right there on those mucosal membrane, and it's going to start to release some of those other enzymes. And those other enzymes are going to, such as the proteases, the mucinases and stuff, are going to start to tear away at the lining, that mucosal lining, and it's that damage in that lining that is referred to as that ulcer or the sore. And depending on how deep that damage can go, it can even get down into the blood levels, and that's when you start seeing the bloody ulcers. So how do you know that you have an ulcer? People will start to have those abdominal pains that you can't relieve that through taking antacids or milks, etc. You may start coughing up blood or having diarrhea with blood. You might have such things as heartburns or weight loss, etc. So how do we treat it? Well, it is a bacteria, so we are going to use antibiotics to treat it. And one thing to recognize is sometimes when they're trying to figure out if somebody truly has ulcers, is they'll do what's known as a urea breath test, because they're looking to see if that urea that's present there, or has the urease, that enzyme, broken it down. But we can also look at blood tests and looking for that bacterium itself, the pylori's presence for it. Okay, so that is the Helicobacter pylori and peptic ulcers, and so that's going to conclude the second video, and we still have one more with the last couple of diseases.